Hello, my name is Mike Andrew Westhoff, PhD, and I'm responsible for WE Research Group at the Research Lab of the Children's Hospital in Ulm. The focus of our group is apoptosis and cancer research. So today's lecture will be on those topics. I hope you will enjoy it. Thank you.
So, of the three pillars of cancer therapy, that is, the three major therapies we've been using for half a century, at least two of them, radiation and chemotherapy, work by mainly inducing apoptosis. Therefore, understanding apoptosis and understanding the underlying mechanisms is essential for understanding how cancer therapy works, why cancer therapy doesn't work, and how to improve therapies in the future. Apoptosis is an ancient Greek word meaning leaves that fall off a tree in autumn. It is, however, not a word coined by the ancient Greeks, as, for example, cancer is, but a word which has been used in cancer therapy for the last 40 years. In 1972, a team of researchers in Edinburgh first described this mechanism and wanted a cool and funky name so they could get it published. Therefore, they went across the street to the Department of Ancient Languages and asked an expert how to call this mechanism. The expert decided, it does look a bit like leaves falling off the trees in autumn, and hence this mechanism is called apoptosis. Generally, we translate apoptosis as programmed cell death, which is wrong. Apoptosis is a program. It is an event that occurs in defined sequences. It is not the outcome. Therefore, it is actually not cell death, but it's the way how a cell dies. So it's programmed cell dying. It is a physiological relevant event. It occurs throughout development and later in life. For example, the formation of digits on a hand or on our feet is dependent on apoptosis. If apoptosis doesn't occur correctly, you get a syndactic hand. That means your digits are fused together. Also, during development, massive apoptosis occurs in your brain. Because, technically speaking, it is easier to kill cells which are too many than to reproduce cells which are too few. So, during development, you develop too many brain cells, unless you're a politician, and then your brain cells die. So if you knock out caspase 9, we'll get to that later, you end up with a huge brain. Furthermore, apoptosis occurs during the regulation of your immune system. Otherwise, your body wouldn't stop reacting to pathogens once they are cleared out of the system. So apoptosis, a completely normal process which happens in your body on a daily basis. And the easiest way to understand how apoptosis works, or the easiest way to understand the reason why apoptosis occurs, is that in a multicellular organism, cells will go wrong. Cells will mutate or will be damaged beyond repair, or will be at the wrong place at the wrong time. In that case, the cell is induced to commit suicide. So basically, the cell dies for the greater good of the multicellular organism. Now, of course, we are in a cancer lecture, so we'll have diseases. And the deregulation of apoptosis is fairly common in many diseases. Basically, there's a whole group of diseases which can be understood as diseases of dysregulation or imbalance. In a healthy body, you have the perfect balance between proliferation, the production of new cells, and apoptosis, getting rid of old and damaged cells. If this balance is destroyed by either increasing the amount of proliferation or by reducing the amount of apoptosis, you get hyperplasia. You get tissue where there shouldn't be any tissue. The other way round, if you increase apoptosis or decrease proliferation, you get tissue loss. So where there should be tissue, suddenly tissue disappears. So basically, the diseases of increased apoptosis that we could think of are listed here on the left side. The diseases we could think of at the top of our heads, where we have decreased apoptosis, are listed here on the right side. I very much doubt that this is a comprehensive list. So if you can think of any other diseases, or you like working with a particular disease, where you suspect or are sure that apoptosis plays also a role, you can easily drop me a line in the forum. So basically, that's why I think apoptosis is important. And this is a brief overview of how you'll spend your next 45 minutes, hour and a half, depending how often you stop the video. Okay? So let's get started with the forms of cell dying. There's a good review which we'll make available online, which gives you a list of specific types of cell death. And depending how you count, there are somewhere between 20 and 50 clearly defined forms of cell death, or cell dying, as I corrected myself earlier. Some of these forms are highly specialized, and some of these forms I suspect only occur when the actual apoptosis regulation is blocked. So they're basically default events which occur when proper apoptosis can't occur. If you use the classical definitions of apoptosis, which are based on morphology, then these forms of cell death highlighted here should be and could be considered apoptosis. Although there's caspase-independent apoptosis, generally we assume that caspase activation is a key feature of apoptosis. Every year, new forms of cell death are discovered and are named. And it seems to be a hobby of scientists to make these names as impronounceable as possible. Again, what we have here is a paper from 2011 where net orthosis is defined. That's a highly specialized form of basically suicide bombing. A cell which is attacked by pathogens dies and shoots out harpoons that are supposed to kill as many pathogens around the dying cell as possible. That is a highly specialized rare event. Nevertheless, it's really cool. If you think about cell death, you should consider it a spectrum, a continuum where apoptosis, as clearly defined, classical, programmed, regulated, normal cell death is at the one end, and necrosis, which was for a long time considered a random, non-energy consuming, dysregulated event. Basically, if the cell can't kill itself, it will just die by necrosis on the other end. Nowadays we know that necrosis, at least in the form of necroptosis, can also be regulated. There is a school which is going back to a more broader definition of apoptosis, where people just call every genetic cell death apoptosis. So every form of cell death which is regulated, which is dependent on certain genes being present and expressed correctly, they would consider apoptosis. The more traditional definition of apoptosis is, as I said before, based on morphology. So next we'll discuss the pathways being activated during apoptosis. As you can see here, it is a very complex mechanism with lots of errors going one way and the other way. And these is just an indication of those pathways that are activated. For each graphic you see here, there's probably at least one error which blocks the whole process at that stage. So this highly complex, rather confusing graphic is already a simplification. However, we probably should simplify it a lot more. Now, the key players, the central regulators of apoptosis, are called caspases. They are proteases which are responsible for the particular morphological features which define apoptosis. So basically, the target proteins that get cleaved by caspases are those responsible for giving apoptosis the particular appearance. In humans we have 12 caspases, and they can be divided in initiator caspases that are upstream of other caspases, or effector caspases that are those caspases that cleave the actual proteins of interest. Caspases themselves are expressed as proforms and get activated by cleavage, either by the cleavage initiated by initiator caspases or by autocleavage in the case of initiator caspases. So a caspase will be expressed in every cell, in every normal cell I should say, as a proform, and when the cell decides to commit suicide, that caspase gets activated and cleaved into smaller fragments, at least one of which is an active protease. The three key caspases you should remember are caspase 8, caspase 9, and caspase 3. Caspase 8 is an apical caspase that gets activated via DES receptor signaling. It is part of the DISC, the DES-inducing complex, and can activate indirectly caspase 9 or directly caspase 3. Caspase 3 is the executioner caspase, the caspase that cleaves the proteins responsible for the morphological changes. Caspase 9 gets activated via the mitochondrial route. That is, cleavage of BIT leads to activation of the mitochondrial DES signaling, where mitochondria break down. You get loss of the mitochondrial membrane potential, the MMP. Cytochrome C and other proteins are released into the cytosol from the dying mitochondria, and cytochrome C together with APEF1 and ATP forms the apoptosome, which activates caspase 9. Caspase 9 then goes on to activate caspase 3. So basically, either you can get DES receptor signaling which is strong enough to directly cause apoptosis, or you get DES receptor signaling which needs to take a little detour and then activate caspase 3. Those are type 1 and type 2 cells. So the activation of caspase 3 leads to the typical morphological changes that define apoptosis. You get membrane blebbing, where basically the cell looks like it's going to explode, but importantly doesn't. That would be necrosis. And then you get exposure of PS on the surface of the membrane. That can be measured with a substance called annexin 5. So once the membrane has basically flipped around and the cell in staining becomes annexin 5 positive, that works as a signal. So basically the PS recruits phagocytes that then eat up the cell. So PS exposure, very early event in apoptosis, recruits the immune system, the cleanup system of the body. Then you get chromatin condensation, where the DNA in the nucleus becomes condensed and then gets fragmented. And then, very late in apoptosis, the actual cell membrane gets destroyed and the cell exposes its innards to the outside world. That is called secondary necrosis. It is basically an event which only occurs in cell culture under experimental conditions, because again, in a healthy human or multicellular organism, at this stage, phagocytes would have already cleared the dying cell from the system. On the lower panel, you see the loss of MMP again, the mitochondria which were loaded with red dye become leaky and the red dye disappears throughout the rest of the time frame. The DNA, after chromatin condensation, gets chopped off into little pieces. Basically, it gets fragmented. And then again, in a different color, you see the late stage where the cell membrane actually is ruptured and breaks down. That should be ruptured. Okay, so let's recapitulate what we have learned so far by a small test. We'll show you some pictures of morphological and biological features of apoptosis, and you have to decide what is actually shown on that picture.